Hughes Medical Institute. Dr. Snapp received his PhD in molecular microbiology from Oregon Health Sciences University, where he studied protein trafficking in Leishmania parasites. So welcome and thank you, Dr. Snapp, for sharing your wisdom and time with our students today. And students, just so you know, you are welcome to jump in and ask a question anytime throughout this session.
Yes, please do.
Yes, okay. So first of all, Dr. Snapp, what is Janelia and what is your role there?
Janelia Research Campus was founded by the Howard Hughes Medical Institute. For those of you that ever watched Leo DiCaprio in The Aviator, that's about Howard Hughes. And back in the late 1950s, Howard Hughes, as a tax dodge, set up a medical research foundation. There was no actual foundation being funded. It was just a way to hide his money. And then the IRS caught up with him, and he decided that maybe he needed to actually have a real medical foundation. And so for many years, the Research Foundation existed solely to fund labs throughout the United States, individual labs at universities. So there's no Howard Hughes Institute beyond there's a place in Chevy Chase where there's a bunch of administrators that work. But back in 2003, there was a decision to have an actual research campus. And they searched all around the country and thought, well, they'd really like to have something close to the Chevy Chase. They'd also like to have something that was near an airport, and they wanted to have something with a lot of space to expand. And so here we are now in Loudoun County on the old Pickens farm, and we were originally Janelia Research Farm, and then we dropped the farm because there were people that kept emailing us to ask to work on dairy science or soil science, which is not what we do. We opened our doors in 2006, and we work primarily on neuroscience and imaging, specifically on different kinds of microscope technology, whether it's for electron microscopy, fluorescence microscopy, imaging animals doing behaviors, primarily related to neuroscience. And so we do a bunch of really cool stuff where we look at how animals do things. Where are the neurons that help them behave? And we have ways of actually lighting up the neurons when they are active so that we can image neurons firing, functioning. And we like to put all of that information together. We have a group of physicists that help create microscopes. We have protein chemists that help put together biosensors that can detect a neuron firing, whether that's the calcium or change in voltage. We have people that have come up with ways of tracing the neurons and figuring out the genetics of the individual neurons, so we know how to actually express special proteins in the neurons that we can shine light on and turn them on or off. We have people that build special probes that can detect the firing of the neurons through the electronic signal, the voltage. We have other people that actually create software and or do higher math to analyze the patterns of the fire of the neurons to figure out how the neurons are interacting with each other and what kinds of patterns are resulting in what kinds of behavior. So we're doing a lot of really cool kinds of science, very Star Trek-y, new agey, cool stuff. We're also starting a new research direction. We're just getting started with that right now, but it's going to be something called 4D cell physiology. And what that means is everything about the inside of cells, how cells communicate with each other, how cells communicate with tissues, and how cells communicate throughout the body. And I'll just put in a minor plug to explain why we are interested in that. Obviously, cell biology is something that's important, has been around for many years, but most of us that study cells, like myself, study cancer cells in a dish on a piece of glass. And that's not a typical cell. Most cells are surrounded by other cells, and they're on a matrix in a live animal. And so what we want to do is come up with entirely new ways of studying cells in their native environment, building new kinds of microscopes, new kinds of probes, new kinds of analytical tools that will allow us to do this. So it's going to be a very exciting new venture that we're going to have in addition to the neuroscience. We have about 50 investigators at Janelia, and that is supported by about 120 postdocs, people that have already completed their PhDs. We have a handful of graduate students. During the summer, we have an undergraduate program where we typically have about 20 students on campus from around the world. And we also have a high school internship. Unfortunately, we do not have it this year because of COVID, but normally that is something that Loudoun County students are eligible to apply for and can work at Janelia for 8 weeks, and they get to typically work in computational lab, doing analysis, doing data annotation, and learning a lot of different kinds of ways in which scientists do research. They get to attend seminars and meet with different scientists at different career stages. That in a nutshell is what Janelia does.
Great, that's awesome. It's very, I don't know, impressive, and I don't know, it's way over my head of what all that's going on. So I admire people that work at Janelia and all that they're doing for our world. How did you get to where you are today?
So I started out in the town of Coos Bay, Oregon. I lived there for the first 18 years of my life. I realized when I was 7 years old, because of my first grade teacher, that I wanted to be a scientist. She had a science table that had skulls and crystals and a terrarium with green anoles, a microscope, a variety of different things that intrigued me. And I was also surrounded by the different creatures at the seashore, or the things that my father would bring home from hunting. And my mother was a nurse, so I was very excited by biology in general. And I knew I wanted to be a scientist when I was 7 years old. The kind of scientist changed a little bit along the way, but usually it was going to be biology. When I was in 9th grade, I watched a special on the show Nova on PBS, and they talked about the history of biotechnology, which was very short history. It basically started in the mid-70s. But they talked about one of the companies that did biotech was called Biogen, and they were located in Cambridge, Massachusetts, and had been started by some people that worked at Harvard. And that to me was very exciting. I literally had no interest in going to Ivy League schools in general, but I wanted to go to Harvard, and I wanted to work at Biogen. Those were my 2 life goals. So fast forward a few years later, and I only applied to Harvard and 3 other schools, Oregon Health Sciences University, Oregon State, University of Oregon, and Carnegie Mellon. And I got into all 4, and I was super excited to go to Harvard. I went to Harvard, and I wanted to work in a lab. And unfortunately, I could not get a lab job. I was offered a few volunteer positions, but I was not making, my parents didn't have enough money for me to be able to be in college without having an actual job. So I worked as a janitor for the first couple of years. And then in my junior year of college, I met a professor couple that needed some help with yard work and some handiwork around the house. So I worked for them. And they had some friends that were in science. And they said, well, we'd like to introduce you sometime. And they need somebody to rake leaves. And I'm like, okay, that sounds good. So I went over to their house one fall afternoon, and there's lots of leaves falling in Cambridge, Massachusetts. I raked over 25 bags of leaves that day, I think. And the wife, Vicky, invited me inside to have a chat and asked me about my goals in life. And I said, well, you know, I really want to go into science. Research is something that's really interesting to me. I might go to cooking school before I go to graduate school, though. And I really did like cooking a lot. And she said, so are you interested in hiring out for cooking? And I'm like, I hadn't really thought about it. But she said, well, how about this? If you want to cook for us, we'll get the groceries. You can cook anything you want. You can eat dinner with us, and we'll pay you. And I'm like, well, that sounds pretty good. So I got to have something better than college dorm food for a while. And I got to meet the family. And so the husband was a professor at Tufts Medical School. And the wife was the head of research at, you guessed it, Biogen. And so at the end of my junior year of college, Vicky asked me what was I planning to do. And I said, I was planning to go back to Oregon and go work for the state parks. And she said, would you like to work at Biogen? I said, yeah, would I? And I ended up getting to work there and made enough money over the summer that I could have an apartment in Cambridge, Massachusetts, which is really expensive, even back in the late 1980s. And I was just hooked. I got to work in protein chemistry, and I spent a bunch of time with a machine called an HPLC, high performance liquid chromatography. That's a separation method for proteins. And back then I was working on one of the early therapeutics for AIDS. So that was very exciting. And I was completely hooked. I knew that I was going to be in biotech, and this was great because in biotech you get paid lots of money, you get lots of shiny new equipment, and you get to help people and you get to develop new therapies. So that was that, and everything was great. And I completed college, and then I worked for another year at Biogen, and then I worked for a while at another institute, Shriners Hospital. They did some connective tissue protein chemistry, and then I had taken a class in college on parasites and was very excited about this. And this is what I wanted to study. I wanted to study microbiology. And I went to Oregon Health Sciences University. I got in there, and there was a really great parasite lab. And during my first year of grad school, I had a horrible realization. I really liked basic research. I wasn't so obsessed with necessarily creating drugs or necessarily curing people. I mean, it's like that's a good thing to do, but it was no longer as exciting to me. I actually really liked the whole idea of basic research and just fundamentally understanding how things work. And that meant that I was going to be an academic. And that meant that I was going to teach and I was going to write grants. And I initially thought that sounded kind of scary. But life has a way of actually leading you to what you're actually good at and what you really do enjoy. And it turns out that I love teaching. I used to teach microbiology lab to medical students in grad school, and that turned out to be a lot of fun. Then I had enjoyed studying parasites a lot. That was really an exciting thing to do. I was studying those Leishmania parasites you mentioned and how proteins are targeted to different parts of this single-celled parasite. Along the way, a bunch of things happened while I was in grad school. And this is actually a theme of my life in science and something that I would like to convey, even if you're not going to be a biologist or other kind of scientist, that these kinds of things will impact your career as you go along the way. And that is, many technological changes happened while I was in graduate school. For example, when I started graduate school, PCR had just been invented. Yeah, I'm that old. How old am I? When I was in graduate school, the largest hard drive on campus in a computer was 64 megabytes. When I was in graduate school, the internet became a thing that was accessible to the general public. Netscape became available, AOL became available. Prior to that, it just really wasn't something that most people could access. When I was in graduate school, green fluorescent protein was cloned. So suddenly, there were a bunch of new technologies which just changed the face of science, literally overnight. In the 90s, so many things changed. And suddenly, my excitement about parasites was now tempered with a love of cell biology and the possibility of doing biophysics, actually looking at molecules inside of live cells. Mm-hmm. And so that's what I got excited about. And I got married, and my very supportive wife said, okay, let's go to the lab where you need to go do your postdoctoral training to learn this stuff. And so I came to the National Institutes of Health in Bethesda and worked for 5 years. I'll answer that question in a moment. Where I worked for 5 years for Jennifer Lippincott-Schwartz, who ironically now also is a PI at Janelia Research Campus. And along the way, I worked on an organelle called the Endoplasmic Reticulum. And that's where all of your secretory proteins are made. So it's kind of an important part of a cell. It makes all the proteins that are on the surface of the cell and all the proteins that are secreted out of the cell. And so I knew I wanted to be a basic researcher. I applied for faculty positions all over the country. I got a job at Albert Einstein College of Medicine in the Bronx, and I lived in New York for 12 years. And then along the way, while I was doing all of this research, writing grants, teaching graduate students, teaching postdocs, and doing research on the endoplasmic reticulum, which I really enjoyed, I discovered another part about myself, that I really love coaching and mentoring young scientists, that people often aren't taught how to do these different kinds of things, how to apply for a faculty position, how to apply for a postdoc, how to prepare a CV, how to interview for a job, how to create a chalk talk that will convince people to hire you for a faculty position. And so I spend a lot of time at Einstein doing these kinds of things. I wrote an ebook on how to apply for these jobs. I coached a lot of friends or people that just came to me and asked on how to be coached on these things, and discovered that this was really something I enjoyed doing. I taught classes, I created new courses at Einstein. I was on the graduate committee, I was on the postdoctoral committee, and I discovered that there was this other side of myself that I really enjoyed doing. And so about 5 years ago, a job became available at Janelia, specifically to come in and take care of the graduate program, to coach postdocs on going on to new careers, to oversee undergraduate program, to have a high school intern program, and to be able to create new kinds of training opportunities to help teach people about all these other kinds of science, or the things that you need to actually run a lab or to move on to your next career stage. And so I've been here at Janelia almost 5 years and absolutely love my job. One of my favorite things are my summer programs. The undergraduates and the high school interns are a lot of fun. introducing them to the ways that science works. They're first times that they get to meet people that are doing incredible science and see what's possible, especially at a place like Janelia, which is just has so many resources and has people with really exciting backgrounds in science. We have aerospace engineers, we have people that used to work at NASA, we have physicists, mathematicians, people that used to work at Bell Labs, people that used to work at some of the top labs around the world. And they're all doing really exciting work and working together. There's a lot of collaboration at Janelia. So it's a very fun environment, and I really love my job now. Okay, I think that's it in a nutshell how I got to where I am.
That was awesome. Thank you. If you want to answer the question.
So there was a question about what's the most transformative technology, and it's a tough call. I mean, my career for the last 20 years has really revolved around green fluorescent protein. It allows you to do a lot of things that were previously not possible, to be able to see cells, to be able to see molecules in cells, to be able to look at variations of green fluorescent protein, which may change color as they get older, or that you can change their color with laser light so that you can optically highlight them and do a pulse chase experiment. That's all been very exciting for me. But if I had to point out what's the most transformative technology, I still would have to say it's computers, because the fast computers allow us to do all the really exciting things we do. For example, the microscopy we do. When I was in graduate school, collecting a simple 5 megabyte image was not fast. The microscopes were slow, and there was no way that we could do some of the movies that we really wanted to do, at least digitally. And now with the super fast computers we have, we can take terabyte movies of 3D, 4D actually, of cells moving in all three-dimensional directions, and we can do it in multiple colors. That would have never been possible without computers. And now we have so many ways of analyzing the data, again, computationally. It's just very difficult to be able to do it any other way. And then we can communicate with each other. We can cross-reference information. When it became when GenBank came out, and suddenly we could see all the different gene sequences out there, that became a huge way of being able to look at whether there were relationships between the phenomena we were studying and the gene we had identified associated with it. It just really changed everything. I'm sorry, I did not see that last question. It flashed by really quickly.
Is there anything special that you need to do research as a job? Anything special that you need to do research as a job? I'm not sure what they mean.
Anything special that I need to do as a researcher to become a researcher? Maybe that's the question. Somebody can clarify. Education-wise.
Yeah.
All right. So I'll answer the first question. So education-wise, it doesn't matter what degree you get so much as you're getting a strong quantitative background. You can learn things along the way. I did not start out as a biophysicist. I started out as a molecular parasitologist. I knew a lot about parasite life cycles, and I knew how to do a lot of gene cloning. But then when I started my postdoc, I started doing biophysics and diffusion analysis. I was able to learn all of that. Once you learn the process of how to explore a system, how to research information, and how to start synthesizing that information, then you really have a lot of ability to move in different directions. The director of our cognitive neuroscience program at Janelia, he was originally at NASA. He was not a neuroscientist working in fly navigation. So you can make a lot of career transitions. You need to learn to learn. That was something that was actually impressed upon me in the first week of college. If you learn the basic principles of how a system works of information, whether it's physics, math, biology, then you're in a really good position to learn other kinds of things. I would encourage you to think very strongly about doing quantitative things, because I think it's easier for quantitative people to learn biology than it is for biologists to learn the quantitative stuff. So I would say, a degree in history probably won't be very helpful, but anything in math, computers, biology, chemistry, physics will put you in a pretty good position to do a lot of different kinds of science.
Wow,
Okay,
they're coming in. Okay.
Yeah.
What is your favorite work experience project that you have worked on from Max?
From which one, sorry?
The student is named Max, and he's asking, what is your favorite work experience project?
Oh, my favorite one. Wow, that is a tough call. I really enjoyed a lot of different things I've done, and I've been doing research since, 1988. So I would say that maybe my favorite one was a project that I did as a postdoc. I was looking through the microscope, and I saw one day that there were these weird structures in the cells. There were these bright structures that we didn't expect to be there. My marker protein, my GFP, was supposed to go just to the endoplasmic reticulum, which looks like a spider web in cells. And it's a very pretty pattern, and it's constantly moving around. But there were these donuts. There were these bright donuts in the cell, and that was very strange. And then I saw it in other cells, and I saw it with other constructs. It wasn't just one different protein that I was expressing in the cell. I saw it with some other ones. And slowly but surely, I started to realize there was a pattern here. There were certain constructs, there were membrane proteins that were making these structures. And I went to talk to a bunch of people about this. It's like, have you ever seen these? No, people hadn't seen these. They thought, oh, the cells are really sick. And then my advisor said, Erik, why are you working on this? Work on something else. I said, I think there's something here. And then I sent some samples to my friend who does electron microscopy, which allows you to look at things at very high resolution, nearly atomic resolution. You can really see the structures. And the structures were gorgeous. It wasn't just a bright donut. There were literally sinusoidal waves, and there were crystalloid structures. I mean, it was really exotic membranes. And so I was like, oh my God, this is so cool. And my advisor was still, Erik, it's just weird. I'm like, no, this is cool. And so I kept with it, and I ended up discovering something that nobody had anticipated. And it also laid the groundwork for figuring out how the cell can make certain kinds of structures through, very simple protein-protein interaction. And to this day, I think it's one of the coolest things that I ever discovered. It was one of those things that everybody told me not to work on, and I persisted, and I learned something really cool along the way.
Went with your gut. I love that. So Juhi would like to know, what do your daily activities consist of?
Well, so my job now is probably really different from what you may be interested in, which is what a scientist does. What I do currently is I meet with a lot of people at different career stages, postdocs, grad students, postbacks, helping them figure out how to go on to the next career stage, apply for graduate school, apply for med school, apply for college, apply for faculty positions. So unless you really want to hear more about that, I'm happy to talk about it. I suspect you want to know more about what scientist does. So as a scientist, at different career stages, the balance is slightly different. When you first start out as a technician, which is usually the first job you have in a lab, it's all about experiments. You spend almost all of your time basically getting protocols, getting asked to do certain things, and sometimes even getting your own project. And so you just spend the days preparing samples, preparing your cells, preparing reagents, buffers, and if you're good, you also read up some papers on your topic, and then keeping your notebook kept up. As a graduate student, you then have to take classes, and you also start learning about some other aspects of your career, and you also do a lot more planning of the project itself, so that you start directing the project. As a postdoc, you're almost always in charge of the whole project. You may talk to your advisor every now and then to think about how to interpret an experiment or how to move in a different direction, but you have a lot more control. And so you plan out the experiments. You think about how you would tackle the problem. I mean, one of the things that people think of scientists doing is just like, you just automatically know what to do. And it's like, look, there are a billion questions out there. You have to decide on what's the most interesting question to work on. And then how do you actually tackle that? Do you attack it by modeling? Do you attack it by collecting chemical data? Do you attack it by looking at it with a really high resolution microscope? There are all sorts of different ways of tackling different problems, such that it's nearly infinite. And so you have to actually figure out what's the best way that you can think of to get the answer, and what's the question you really want to answer. But you do a lot of experiments, collecting of data, thinking about it, reading a lot of papers, but you also go present your work at meetings. You may present your work for lab meetings, but you also would write papers. You also get invited to give talks, get invited to also go to courses where you might learn some special techniques, and email a lot. I spend a lot of my day emailing collaborators, emailing people to make sure that some experiment's getting done or that some reagent is being sent to me, that my paper is moving along in the pipeline for getting reviewed by an editor. A lot of emailing. And then when you become a PI, a group leader, a professor, you have an additional component to all that. You may do some teaching, but you also do a lot of management. And I didn't say anything along the way about learning how to be a manager, and that's actually one of the reasons that my job exists, is to help postdocs learn about management, because when you become the head of a lab, you don't have to manage people. You have to actually motivate them, convince them that the research projects that you're doing are something that's exciting for them, that they will want to do it. You have to resolve conflicts. If two people want to work on the same thing, how do you find a happy medium for both people? And I also have to manage a lab budget, and you have to write for grants, and you have to help everybody learn how to write papers. You get invited to go give talks, you get invited to meetings, and then you serve on a lot of committees. I served on a bunch of thesis committees for students. I served on faculty committees for, the animal use committee, the non-human radiation safety committee, all these different kinds of things. It's very busy. There's a lot to juggle, but you get to do science. You get paid to do science. You get paid to solve the answers to the questions that you're interested in finding out about, and that's why it is so rewarding.
That's awesome. We're getting a few more questions. So Nitya would like to know, what are some things you recommend for high schoolers who are interested in biology to take part in or to look into?
So you're blessed by being in an area where there are opportunities for high schoolers. When I was in high school, there were no lab jobs in Coos Bay, Oregon. So you could apply for our internship. You could apply to work in labs at the NIH, at Georgetown, at George Mason, at George Washington. There are a number of different places that would potentially entertain having high school students in the lab. I had high school students in my lab at Einstein, and they were really fun to have. So that's one opportunity. Another one is to just get other kinds of experience, whether it's going to work in a computational summer program or a computational lab. Getting experience doing different kinds of lab things is great, even for a biologist. Even if you're going to be, the ultimate cell biologist, I'm pretty confident that getting a lot of experience coding or getting experience learning about computer vision or something like that can also be very useful, or even doing something in chemistry or physics. It's more about understanding how science works, and the more you see how to solve problems from different scientific perspectives, the better kind of scientist you can be.
Okay, awesome. Along that line, Ari would like to know, what do you think the future of cell research will look like?
I think it's a really exciting time. I think that we've been doing the same kind of cell biology for the past 10, 15 years. It hasn't changed dramatically. We can do higher throughput, so we can look at a lot more cells, and we can do things faster, and we have more tools for looking at cells, new microscopes, new fluorescent protein biosensors that tell us whether there's a flux of calcium, whether there's a change in pH, whether there's where a protein is or it's being turned over. But as I mentioned, with this 4D cell physiology, which I do think is the future, we're going to be able to study cells in their native context. We have to do that. The amount of oxygen that a cell normally receives is much lower than what we have in a tissue culture flask. It's very low, and it's been shown that the gene expression in a cell in low oxygen is very different. The environment that the cell interacts with, the molecules in the matrix, the cells that the other cells interact with, that all influences how the cell computes. And we can't get that kind of information from just growing cells on a glass dish. And then I think the other component of all of that is an even deeper, more powerful computational analysis of cells, that we're getting to the point that we have the computing power to start modeling, pools of individual molecules, all the different kinds of chaperones, cytoskeleton proteins, enzymes, transporters, all these different things within a whole cell. And we can start to ask questions about, are our models correct? Or do they break down when we start to have all these different processes? Is one process taking over, or can they all coexist at the same time? I mean, as biologists or as imagers, we normally only have a couple of tools for seeing a couple different molecules or structures at a time, and yet all the things are happening all the time in a cell. And so how is all of this regulated? How does the cell make decisions? We just haven't had any way of really looking at this problem in a comprehensive way. I think those tools are now about to be in our hands, and it's going to be exciting to be at Janelia because we're going to develop some of them here.
That's awesome. Okay, let's see. Sona, I hope I'm saying that right. She asks, or he asks, what are some tips you have to retain and memorize a lot of information?
Okay, so I took a speed reading class my junior year of high school. It was only about 2 weeks, but it was really helpful. One of the more helpful things I've ever done in high school. And what I learned was how to focus on the important information in a paragraph and how to read a lot faster. Because one of the problems you're going to have is that there's just so much material out there. To be able to get to the heart of a paper, a research paper, and know what are the things to focus on, what are the takeaways, and how to learn to critically read them, because some of them you can't skim through it. You actually have to critically read it. And so to know where to focus in the reading of that paper becomes a very powerful tool. Memorization is a funny thing. So I think a lot of people think of scientists as memorizing everything, being an encyclopedia. And to some extent, that's true. I honestly believe that the more that you know in terms of factoids, the easier it is for you to imagine the ways that different pieces might come together, because you can think of different kinds of scenarios. You've thought of those things. With the availability of the internet, it is possible to look things up on Google, but if you've never heard of something before, it's much harder to look something up if you have no idea what it is. And so I think at some level you need to learn, how these systems actually work and the things that are relevant to what you do. I didn't spend a lot of time cramming information, but there's definitely a component of that. I think a lot of schools are getting away from that. That is old school. These days, the idea is that you learn how to synthesize information and get like a couple of very explicit examples, and then you can translate that. But at the end of the day, I would encourage you to go to seminars, read papers, talk to scientists in different fields so that you get exposed to a lot of different things. At the very least, that helps you start to appreciate the kinds of questions you can ask and expand the ways that you might think about a problem rather than being constrained. The other thing I would say about memorizing information is sometimes the information is wrong. And one of the things that's very important to appreciate is how much in flux science is. Even though you have textbooks and there are even published papers, things change as people get new technology, new ways of measuring things. And so what was once accepted wisdom, and realized that was just because of the way that we measured that sample, that in fact it really works quite differently, or there's many more components to it. Something that was just simply an arrow in an equation turns out to have all of these additional steps. And so I think the useful thing is learning the parts that you're aware of, but being fully flexible on the idea that new information may become available and you need to change how you think about something and not think about it statically.
Yeah, good point. Okay, Venkat would like to know, how do you pay and funds work as a researcher? Is it a stable job?
Okay, there are different kinds of researchers, and this is a great question. So if you go into industry, you tend to get paid better. Payment is because you either had venture capital investing in your company, because you're a big pharmaceutical company and you have other products that fund your research, or maybe because you've started selling products and your company is able to fund this. In academia, how you are funded varies. It's typically through grants, and so you write grants multiple times a year to be able to fund your lab. Sometimes it's money directly for your salary. Sometimes it's money for the people in your lab, for the reagents in your lab. If you work with animals, to pay for mouse care, that can be very expensive. Or to write a grant for a big new piece of equipment. Like I wrote a $400,000 grant for a microscope, a very fancy microscope. And so, it's usually through grants or through industry. There are also people who work for the government at the NIH, for example, and so that's actually funded all by tax dollars. In terms of stability, so that's actually an interesting question. If you ask most people, they would say that it's safe to be in industry, because in industry, it's not reliant on writing grants, and so it's very safe. And that's not really true. The reality is that companies change projects. They decide that they no longer want to work on something, or that there's a technology that's dead, or that somebody else has made an advance and that you're not going to be able to compete. Those are all business decisions. And so projects just end. And that can be either you suddenly start working on a completely different project the next day. I've seen this happen. Or it could be something as harsh as, actually, that we're not going to keep you. We don't need your expertise anymore. That happened to some people that I was collaborating with when I was at Einstein. We did some work for Merck, and we helped them collect data. And at the end, they decided that they weren't going to continue the project. And the 6 people that I was collaborating with, they weren't at Merck after a few months. In academia, people are worried about writing grants. That is something you might hear professors talking about. Most people have a fairly stable job in academia, and especially depending on the kind of place where you are. At medical schools, you have a lot more responsibility for writing grants because so much more of your salary and your lab, whereas at universities, you do teaching and colleges. And so that pays for a lot of your salary, and the grants are more about just for running the lab. So there's some variation there, but I would say the stability in science actually isn't bad. People may look at it and say like, oh, well, that's really tough. But I would say that the thing that's worth looking at is how many people are truly stable in careers today. Everything that I'm aware of is that people change jobs every 5 to 10 years. And so I wouldn't think so much about having a career set where I'll be at the same desk for the next 20 years. Those careers exist, but in many ways, things are just much more flexible because technology changes, new opportunities arise, new kinds of businesses become available. There was no social media when I was in grad school; it didn't exist. So I wouldn't worry too much about stability. I would worry about doing the things that excite you.
Awesome. Okay, Rachel would like to know, how might things change in the world if another breakthrough in cellular research were to occur?
So many different ways. All right, so the stuff that we're doing right now for the COVID vaccines, all of that is basic molecular and cell biology. The idea of using RNA as a vaccine. Before this research by a very unsung female scientist, everybody made vaccines by killing viruses or killing bacteria. And then it became possible to sometimes express a gene, a single protein. You could purify the protein. And then, this scientist had this idea, what if we actually got the code for the protein into the cells and had the cells make the protein? And it turns out that it works great. These new RNA vaccines for COVID are the first RNA vaccines ever used in humans, and they are amazing. Vaccines are considered good if they have better than 75% efficacy, and these have 90 to 95% efficacy. It is off the charts in terms of immunology. So that's one example, but there's a number of other examples. So there's CAR T therapy, which is used for treating cancer. It's actually training immune cells to hone in on cancer cells and kill them. There is the whole area of tissue engineering so that you can make replacement organs. We're going to have those in our lifetime. People have started making these things, and you may be able to replace a liver or a pancreas or something else that people need. I think the other aspect of it is just the basic fundamental biology. If we start to understand how cells communicate, how cells form organs, or how the brain works, then that could also have spillover effects, broader impacts. If we understand how the brain works to even some degree at the cellular level, that might influence how we design artificial intelligence.
Wow. Okay, and Max would like to know, what would an internship look like and what would you do?
So high school internship, I assume the question is. So we've had 5 years of interns from Loudoun County schools. And what they do is they come in for 8 weeks. There's usually between 5 to 8 or 9 students. They are assigned to different labs. And they typically either do data annotation, they do computer vision training of the computer vision program to distinguish different kinds of behaviors of an animal, for example. Some people who have more advanced math and computational skills have done machine learning analysis of algorithms from the neural spike patterns, or to figure out how to trace a neuron, do some modeling of whether you can think of a neuron as having multiple outputs or having a single output. There's been a variety of different projects, and it really depends on your level of math and level of computation. The little dirty secret is that if you are doing Python or MATLAB, you will have a leg up. We also like people that are doing C++ and R, but we don't really use Java for most of the kinds of things that we do. So if you have more experience doing Python or MATLAB, and to some degree R or C++, that will put you in a much better position to be able to do some of the kinds of things we do in our labs. And then the students would come in 5 days a week, 40 hours a week, doing data analysis, data annotation, etc. We have seminars that they can attend. I host a weekly lunch with scientists at different career stages where you can learn about how people got to where they are as a grad student, a postdoc, a PI. We also have an end of the summer presentation that you give. We have a barbecue that you can attend with your PI and your family. Students also get involved in some of our activities after work. Sometimes we have races, or we have just a happy hour or a seminar that people attend. We have book talks. Yep.
Wonderful. So do you anticipate the internship, or you hope that'll be available in 2022?
That is the expectation, yes.
Okay, hope so. Okay. Victoria would like to know, what's the difference between the high school and undergraduate internship?
Quite a bit. So the high school internship is only available to Loudoun County students, and it's a daily internship. You do not live on campus. Sorry. And so you drive to campus, or your parents drop you off. The Janelia Undergraduate Scholarship is a 10-week program. It is international. We take students from all over the world, and they work in individual labs. They work all hours of the day because that's what they like to do. And they live together with the other undergraduates, usually in a group of 8 in each of the houses. We have evening journal club sessions, as well as evening sessions on presentations. We have an end of summer poster presentation. We have a couple of barbecues. I take students out hiking, bird watching on the weekends. People for 4th of July sometimes go up to New York City, go see the fireworks, just experience American culture. So it's a much more intense experience. But we have a number of people that do the undergraduate program that are actually local or are here in the U.S. as well.
I'm just, for my curiosity, how many of these internship students or interns do you actually hire?
Hire for the internship?
No, hire after they've graduated from college to you, they actually start working for Janelia.
So we've only been in existence since 2006, and we've only had 5 years of high school interns. We've had the undergraduate program since 2008, and Jan Funke, who was a undergraduate intern in 2009, is now one of our group leaders.
Okay, awesome. Do you know how many students that graduated through Loudoun County Public Schools, over all the years that work at Janelia? I know the one that we talked about, but do you know the background of very many of the students, or the
postbacks? The postbacks less so. So postbacks, again, are people who've graduated from college but have not gone on to graduate school. Not that everybody goes to graduate school.
Right.
So I would say it's not a small number. I would say it's in double digits. We have things like the Connectome, the CAT team, and there are people that come work, in some of the computational labs, or they work in the vivarium where they take care of the animals, people that work in the fly tracing or fly management group. So there are several local people that didn't come from out of state to work at Janelia, but plenty of international people as well.
Right, I guess that's what I was asking. We've got just 1 more minute. Anyone else have a question? You can ask Dr. Snapp. What are the applications of computer science in cellular research?
So many. So right now we have people that are segmenting cells. So they've actually done 3D reconstruction of the electron micrographs from using an electron microscope, and they're trying to figure out ways to automatically segment them. They want to basically automatically assign, that's the endoplasmic reticulum, that's the nucleus, that's the Golgi complex. Because we've got a whole fly brain that we recently reconstructed, and that's 100,000 neurons plus at least as many glia. And we don't want to have to individually go in there and say, that's where all the mitochondria are and all that. So one of the things is just being able to do that. Computationally, the other things are analyzing high resolution data, or coming up with ways. We had a seminar just last week on how to actually take images and make them better by using computational algorithms to basically apply different kinds of transforms to the image data, as well as ways of looking for patterns in cell behavior that we can't normally detect with our simple human brains and simple eyes. There's only so many axes of information that you and I are able to easily digest, whereas with higher level math and computation, it's possible to think of hundreds of axes. And we need to do this now. We need to be able to start working with the amounts of data that we can generate. I saw somebody had a question about where can we get more information. I assume questions about the high school internship. This is announced by Loudoun County High Schools. You should get announcements at the beginning of the year and when the actual application is available. I have a website at Janelia. You can go up and look under Janelia and U, Y-O-U. And there's a thing about the high school internship. There's something in there about the application. Do not send me an application. It will be rejected. The application is not meant for me. There is a thing there for applications for people that are Loudoun County residents that aren't attending the Loudoun County Public Schools, people who are going to Thomas Jefferson, or people who are attending some of the private schools here. They send their applications to me. You do not. You send your applications into Loudoun County Public Schools, who actually then sort through the applications. But that's where the information is, and you can talk to students who are former high school interns, and they can tell you about their experiences.
That's awesome. Well, thank you so much. That's the end of our time, and I just greatly appreciate your time and your expertise and sharing with all the students. I know they've learned a lot. I certainly have.
And if you have more questions, you're more than welcome to email me. I'm happy to entertain your questions. I hope you all have a great day.
Thank you so much. Take care.